Hi, this is Jim Wells again. I'm going to tell you about drug discovery part 2, from hit to pill. Now, last time we left off, we had taken you up through the stages of these 9 stages. We'd taken you up from the target identification stage to hit ID, and now we're at the hit the lead stage. And the key thing about this stage is that we want to identify compounds that don't just bind to the protein, they actually work inside a cell, and they actually show selectivity in a cell. And this is yet another elevated level and gets us closer to what we want, which is a drug that's safe and efficacious in people. The key aspect of hit the lead stage is an iterative process in which we not only show that the compound works in a biochemical assay, but we also demonstrate that it works effectively and selectively in a cell-based assay. So it can actually go through the cell membrane, reach the target inside the cell, and if it is an intracellular target, and engage that protein in a cell-based assay. So in starting this process, the compounds start off with potencies that are weaker than we would like, as shown in this biochemical binding assay. What we're looking for is compounds that we make through medicinal chemistry that will improve the potency of the hit compound at least a factor of 10, ideally a factor of 20 in the biochemical assay. We'll also be looking for things that start off from the hit stage that have weak cellular potency, shown here, but with medicinal chemistry that correlates with the biochemical potency above, drives the cellular potency to be more potent in a cell. And this is all toward, the goal here is to get potent compounds that are cell active. Now, we also look from the stage for several other important properties. For instance, we don't want it to bind to other off targets that are related to it that may cause toxicity. So what we prefer is that the compounds have potency that are at least 10-fold weaker to the closest related target. We'll also be looking to see that there's chemical evidence that we can advance these compounds. When you look at compound structures, you should see that there are compounds which both affect the target and compounds which don't affect the target that are fairly closely related, suggesting that they're binding to a single site, because one of the properties of a drug is that it binds to a single site on the target protein. We'd like to know the molecular target. Michelle mentioned that you can start off these hit-finding expeditions just looking for cellular activity. In that case, we don't really know the target that's involved. At this stage, we really would like to have a molecular understanding of what that small molecule is engaging. Lastly, and very importantly, and this is especially for formulations reasons, we look to have compounds which have solubilities above 100 micromolar. And that's because we wish the compounds to be soluble and dissolve well once they're administered to an animal. Okay, having successfully passed this stage of the hit to lead stage, we're now into this very important and pivotal stage called lead optimization. This is where we're looking to see that compounds that we've created can actually work in a whole animal, can reach the protein target through a cell and through the circulatory system of the animal. Okay, so I think at this stage it would be good to understand what happens when you swallow a pill. Okay, here's our patient right here, going to take a pill. There are the pills. They go down through the saliva into the stomach, where they're subjected to pH 1, so they have to survive that. They come out of the stomach and they go straight into the duodenum, which is this arrow here, which you see goes straight into the intestines. Now, the intestines have about 200 square meters of surface area to absorb. That's about the size of a tennis court. There's a lot of opportunity to absorb a drug, but it has to have the right physical properties to get across that intestinal barrier so it actually enters into the bloodstream. Once it does get across that in through the intestine and into the bloodstream, the first thing that it does is it goes straight into the liver through the portal vein here. And then it meets a series of very important obstacles. they include a variety of enzymes that are in the liver that are meant to detoxify and get rid of these foreign small molecules. So they would be things like P450s, which oxidize the compound, or hydrolases, proteases, lipases, esterases that would be hydrolyzing compounds. They would be glucuronidation enzymes and the like, which tag them so that they can be rapidly excreted. Of course, all of these things can reduce the potency and the availability of the drug to have its effect in the peripheral tissues where it's probably got to act. So it really does, the drug has to get through this very important gauntlet here of the liver in order to go on from there throughout the circulatory system, first into the right atrium, then into the lungs, and then back from the lungs into the left atrium, and then through the arterial system, all the way throughout the body. So understanding the pathway that a pill has to take is actually very important in this drug discovery process, because even though a compound can be great in a biochemical assay or a cell-based assay, if it doesn't work in this system over here, then it's not going to be, it won't be a drug. Okay, once, now understanding that, we then, and having successfully passed the hit the lead stage, we're now ready to do lead optimization. And that again is an iterative process of chemistry and biology paired together, but this time at the animal level. So the very first thing that happens when you have compounds from the hit the lead stage is that you'd want to determine how well do they survive in the body, how long do they live in the body. And to do this, you typically give either an injection of the compound or feed an animal this compound, and then determine the PK of the compound. In other words, how well does that compound, this is time along the x-axis here, and amount of compound that's in an animal along the y-axis. And as the compound is injected, you see that there's a rise in the compound levels as it goes into the circulatory system, and then it decays. And this half-life and other parameters are used to judge how well will that compound work in an animal, or how long it will last in an animal. The next stage is called the pharmacodynamics part of this, and there we're actually looking to see how active is this compound in an animal model. This I show here as an example of a xenograft model for cancer. This, is, for instance, is time along the x-axis, and along the y-axis is tumor volume. You can see if the animal is untreated with your drug candidate, the tumor grows rapidly. That's this blue line here. And then in the case of a compound that shows some efficacy, if treated with the animal, treating the animal with that, you can see you can suppress the growth of the tumor. That would be a good result. Now one would continue along this process of lead optimization until one has gotten compounds that show good PK and good animal efficacy according to those guidelines we discussed at the beginning of this lecture. The notion of lead optimization is really one of trying to push compounds from the vast amount, trying to identify compounds from the vast chemical space that's out there. Through the hit the lead process, we've identified potent compounds, both biochemically and in cell assay. We use pharmacokinetics to identify those compounds that have both potency and good pharmacokinetics. So we take compounds within this area of the Venn. And then using our animal efficacy experiments, our PD or pharmacodynamic models, we're really interested in the compounds that are sitting right in this narrow area here. So it's this iterative process of testing these compounds in pharmacokinetics, pharmacodynamics, and cell and biochemical assays that ultimately then identifies a compound that can be deemed a clinical candidate. This would be a compound that would be ready for IND-enabling studies and hopefully on the pathway to being a drug. So the goals for an oral drug then, after this process, again, are that we're looking for compounds that can be dosed once a day at hopefully less than 100 milligrams per day. We want reasonable protein binding potency and reasonable cell activity. We want a decent half-life so that it only has to be taken once a day, and we want good oral uptake so that it can be taken orally. Okay, all of this work I told you about, this reminds me of this, this Greek myth of this character down here who you can see is exhausted, and that's how you feel once you get to the clinical candidacy stage. You've just spent a lot of time, a lot of effort from target ID, hit delete, and lead optimization. You've just reached this plateau here, and now you look up and there's all of this stuff still to go, because that's the drug development stage that you need to get to finally reach the nirvana. So now, having done all the work in the drug discovery part, which is basically this area from target ID through lead optimization to identify a clinical candidate, that typically takes somewhere around 3 to 4 years to go through that process. There's still a whole lot of work to go on, from IND enabling all the way through registration and FDA approval. This process is a much more expensive and longer process, about 4 to 8 years to get through that process. And it's worth considering what are some of the reasons that compounds, we show this as a funnel because there's a lot of attrition that goes on in this process. In fact, when a compound enters the clinic here in phase 1, only 1 in 10 compounds will actually make it to being a pill. What are some of the reasons that they don't make it all the way to being a pill? About a third of the reasons are due to lack of efficacy. So this is when a compound shown to be efficacious in animals and all of the target ID experiments that were done before, this really reflects the fact that we didn't have a validated target to begin with. That there's more going on in the biology than we thought, and it's sort of back to the drawing board about what is actually driving that. Other reasons for compound failures, about a third of them die because of toxicity in humans. These are things that we couldn't really model in animals, and so they wind up being disqualified, as they may have failed in phase 1. Other reasons for failure are pharmacokinetics. They just don't have the right clearance and the right properties to be once a day or twice a day kind of medication, and so they can fail for those reasons. There's another and much less likely failure, which is due to commercialization reasons, and this may be because the company just feels that the market size is not what they thought. The drug is really not going to be worth developing further, and so they will stop that. There are many other reasons that might happen, but these represent the lion's share of the kind of failures that happen in the clinic. So we really need to get much better at doing this. We know that healthcare costs are spiraling out of control, and this represents something like 15 to 20% of the GNP. And new drugs and new therapeutics can really spare us a lot of money in our economy if we're able to find new compounds that can ameliorate a disease. Because it's much better to treat it early than to have to deal with the symptoms and the ramifications after. This also highlights the fact that we need to get to prevention and cure versus crisis and symptom, which is the process that we're in now. It's much cheaper to prevent and cure than it is to manage a crisis or just simply manage the symptoms of disease. Now, how are we doing in this process? Well, there's something like 20,000 gene targets, meaning 20,000 genes that encode for proteins. If each of those were associated with a disease, there's not a one-to-one correlation, but you could imagine that we should be able to find a lot more drugs, because currently there's only 200 drug targets that are addressed. And that means we're a long way from really saturating the genome, or the proteome as it were, with potential compounds that can affect disease. The other thing is that these days we're only adding, the FDA, while it approves something like 30 to 40 new drugs a year, we're only adding 3 to 4 new targets addressed per year. And that's because many of the drugs that are approved these days are what are called Me Too drugs. They're second generation drugs that may have better pharmacokinetics, less toxicity, but they're not really addressing a brand new mechanism. We truly need to address new mechanisms if we're going to affect our health care. So in the last little bit here, I wanted to tell you why I think drug discovery is so exciting from a scientific point of view. And I'm going to do this, these next few slides with the example of Gleevec. Gleevec's a new drug to treat CML and other cancers. And this was discovered from some very careful molecular biology and oncology studies that in genome-wide association studies that showed that this target, the ABL kinase, gets hyperactivated when it's fused in patients with CML because of this fusion with this protein BCR. So BCR-ABL, when they are fused together at the gene level and then encoding a protein, leads to this hyperactive protein kinase that phosphorylates its substrates much more active than it should, and that drives cellular proliferation and chronic myelogenous leukemias. Now, turns out that this is a kinase which binds ATP, and using high-throughput screening, a group at Novartis was able to identify compounds that would actually displace ATP from BCR-ABL and thereby inactivate the kinase so that it would no longer drive CML. And this was a spectacular discovery, one of the first really highly targeted kinase inhibitors that showed dramatic effects on this really life-threatening disease. Another aspect of drug discovery that's so fascinating is the fact that it allows us to understand how small molecules bind to their proteins of interest. They use weak forces to carry out this binding reaction. And it's those weak forces that if we understood them better, we could really make better drugs, discover drugs more rationally and more completely. And so by looking at the structures of compounds bound to, here we're showing Gleevec, bound to BCR-ABL in the spheres here, we can begin to understand the hydrogen bonding interactions between the drug, the ion dipole interactions between the drug and the protein, and hydrophobic interactions between the drug and the protein. And we can make analogs, chemical analogs of these drugs, and see how that affects potency, so we can really understand the molecular details of how these contacts drive potency. And in that way, we can then understand how we can build better ones, because by seeing the values of certain interactions, we can encode those into the small molecule to drive potency. Another aspect that I find particularly fascinating is that proteins themselves are quite dynamic. They're moving in solution. Their side chains are rotating on the surface of them. This is our friend BCR-ABL. And when this protein binds its substrate, it undergoes conformational changes. And when it binds the drug Gleevec, it involves other conformational changes. So maybe if we could show this movie here, you can see here, as the protein binds the substrate, there in this green region, you can see a conformational change. And then when it binds Gleevec, there's a much larger conformational change that ensues. These conformational changes are very important in understanding how proteins bind small molecules and how they can be controlled, because they are moving dynamic objects that can be trapped in different conformations, and drugs allow us a way of studying these kind of phenomena. Now, another thing that's very exciting about drug discovery is it allows one to have a chemical biologist or biologist to have tools that can allow you to affect a biology in a cell in a very rapid and dose-dependent manner. Small molecules will act often on the time scale of seconds to minutes, and this is much faster than such probative technologies like knockout mice, which take the development of the whole organism, or siRNA, which typically takes 72 hours before you've had a complete knockdown of the protein of interest. Another aspect of drug discovery that's very exciting, and this is for the pharmacologists, is that it allows them tools to understand, to relate the physiological effect of inhibiting or activating a particular target on the effect on the animal itself, and in how that relates to the disease. And so this really provides important tools for pharmacologists, and has for years, for understanding how particular biologies are regulating the phenotypes that we see in animals. And lastly, it provides tools for geneticists and clinicians to relate pharmacogenomics and epidemiology to disease. As I mentioned, the ultimate target validation is a drug itself. An approved drug that is shown to be effective in a particular disease indeed validates that target, that is important. And additionally, by looking at responders and non-responding patients and the difference in their genomes, we can begin to understand what other factors modulate their, the disease itself, and what makes them sensitive or resistant to certain compounds. And finally, one of the things that's so great about drug discovery and development is that it provides the fundamental basis for a healthier society. Drugs have made a huge impact on our health, and will continue to do so. We have a lot more to do in this area, as I've shown you, this is something that I think is particularly exciting and worthwhile doing. So I'd like to thank you for listening to this presentation, and see you later.